Good morning and good afternoon, everybody, and welcome to this Evosep webinar with Thomas Angel from GSK. Let me start with a little bit of practical information. So as you can see, we have set up this webinar as a GoToMeeting. Everybody will be muted throughout the presentation, but please feel free to add any questions in the chat box, and then I will ask these questions after the presentation. So I would also like to extend a warm welcome and thanks to Thomas Angel. Thomas Angel is currently a senior scientific advisor in the Exploratory Biomarker Assay group at GSK, developing protein turnover assays for bespoke or tailored targets through the combined application of stable isotope metabolic labeling and high resolution mass spectrometry. So the title of his talk today will be Targeted Assay Development for Quantifying Protein Half-Life over a Range of Mouse Tissues. So without further ado, I will give the word to Thomas, and I'm really looking forward to your presentation.
Well, thank you so much. It's a real pleasure to have an opportunity to share with everyone today some of the exciting activities that are ongoing here at GSK. Hopefully everyone can see a pointer and the audio is clear. And with that, I'll get underway. So today, I'm going to be telling you about this what I think is a very powerful pairing of technologies. So an analytical approach and ultimately application of these technologies for targeted quantification of protein half-life. So the main strategy that we're using is an in vivo metabolic labeling approach, which enables protein turnover measurements. So that the significant elements of this approach are that and relevant here are that we're coupling short LC separations with high resolution mass spectrometry, and together these tools are enabling the quantification of protein fractional synthesis over a range of mouse tissues, and basically the short separations really make these activities very feasible in the group that I'm in. So the parts of the workflow that we'll be discussing and I'll share with you today are try and describe to you what the metabolic labeling entails when we're using D2O as a universal stable isotope labeling reagent. Very briefly, the pairing of the Evosep 1 LC front end, which makes designing and executing using very short LC separations very feasible and tractable, and it's a very robust system on the front end, which is very nice. And I'll describe the targeted assay development that we're using for protein quantification for the fractional synthesis measurement itself. So when we so here's an overview of measuring protein turnover, and this approach can be used in cells in culture, in preclinical animal models, and in human beings, and that's what's really exciting about it. So you have a stable isotope labeling strategy that can span from the simplest preclinical experimental system to a clinical context. So you're using the same tracer, you're basically going to be using the same analytical tools, so that translation from preclinical to clinical activities is relatively seamless. And it's pretty exciting to me to be able to think about doing stable isotope labeling in humans as the same way you would with a Silac mouse. And it's really as simple as sending people away, having them drink deuterium oxide which is shown in bullet point 1. At some time later, sampling proximal body fluid. This can be urine, blood, saliva. From that proximal fluid, we would measure the exact deuterium oxide enrichment in the total body water of the system. So this can be cell culture again, preclinical animal or human. And at a relevant time point, we would take a tissue biopsy or collect a biofluid of interest, isolate the proteins from that biofluid of interest, and measure using a bottom-up shotgun proteomics approach, proteolyzing proteins, measuring the peptides generated, and very carefully quantifying the isotopic profile for each peptide that's identified. And what we measure is a shift in the isotopic profile as a consequence of deuterium oxide incorporation. So as more neutrons are added to the biopolymer, you get a shift from intensities in the monoisotopic mass, M0, to increased abundances at higher masses in the isotopic envelope. And it's this shift that we interpret as the label incorporation, and that is used as the readout of new protein synthesis. So very simple question or statement, and this is true, that changes in the production and clearance of any protein of interest precede changes in total pool size. So that's important to know. So what I'm really trying to get at with the title here is that we should be able to measure changes in fractional synthesis earlier than and make a meaningful measurement earlier in time than we could necessarily measure changes in pool size. So it's a quicker, can be a quicker measurement. You look for the biological system to evolve over a shorter duration of time. And importantly, we're only measuring the biological processes that are occurring in a very defined window of time. So that means that we don't have to worry about all of the historical biology that's gone on prior to, say, if we're looking at the consequence of disease process or a drug intervention or disease modification. We can very temporally dissect our treatment effects or drug effects. And importantly, the measurement is, because we're looking at the isotopic profile distribution, the way we do the analytic, it's normalized. So everything is normalized to itself. And that removes technical variance, which is really quite exciting. And the utility of measuring protein flux, we get a kinetic rate constants, the K and the half-life of the biopolymer, a protein of interest. Knowing that half-life allows us to accurately synchronize PK/PD readouts, and it also allows, applying this technology allows us to characterize disease phenotype and treatment phenotypes for that matter. So it can be really quite impactful. So how does deuterium oxide get incorporated as a stable isotope label into proteins? So deuterium oxide mixes rapidly with the total body water of the human preclinical animal. Once that equilibration of water throughout the organism occurs, water gets incorporated into amino acid side chains during de novo synthesis. And this is a non-reversible label incorporation. So once deuterium oxide is incorporated into the amino acid side chains during de novo synthesis, it does not back exchange with bulk solvents. So the label is persistent. Then obviously amino acids get incorporated into proteins during new protein synthesis. The number of deuteriums that gets incorporated into any amino acid is predictable and reproducible. And this has been initially observed by Comerford et al., where they used tritium to label mice, and has been confirmed in more recent studies with humans and mice and cells in culture and things like that. So that gives us, if we know the amino acid sequence of a peptide, we know how many sites could be labeled. We can accurately model what the isotopic profile would look like if you have 100% new or 100% label incorporation into any peptide. And so just as a simple demonstration of what we're looking at is if we have protein at some time 0, we have all unlabeled protein. When we sample from that pool of protein and look at the isotopic profile of a proteotypic peptide, we get a distribution of intensities, of normalized intensities of M0, M1, M2, and so on. When we have some, say 25% new protein produced over a time interval, what we see is we should see a loss of M0 and a slight increase in the other heavier mass isotopomers. And this, is persistent. This change is consistent over the course of synthesis, and it fits a rise to plateau model, this fractional synthesis. And so we leverage these relationships. If we look at the proteome in its entirety, there's a range of protein fractional synthesis rates or protein synthesis rates. And so when doing an experiment, it's important to understand the turnover rate of your target of interest, and that will help guide you in designing those experiments. So obviously, for very slow turnover proteins, which deuterium oxide is very, very is ideal as a labeling tool, where you have very long-lived and very short, very slowly synthesized, very slow fractional synthesis. Deuterium oxide really performs best because the label, you can dose people, animals. It has a relatively long half-life. You can keep people on fixed label exposure for weeks to months with no toxicity or adverse events, and what you'll see is just a brighter and brighter signal as you go out in time for labeling for days to weeks to months. Now, to the really exciting part, as I see it, of this talk today, where we're looking at a more traditional approach to metabolic labeling with deuterium oxide would be a discovery kind of experiment where we look at peptide level data and the mass isotopomers and the shift in mass isotopomers. More recently, we've developed a technique where we are basically generating or have created targeted assays, where we are using, instead of a peptide level signal, we measure the stable isotope incorporation into individual amino acids within a peptide, where it's a MS2 experiment. And what we get is a very, very large change in the isotopolog abundances as a function of metabolic labeling, which increases our accuracy and our sensitivity for measuring changes in fractional synthesis with low body water exposure. So this is an ideal implementation when we're moving from preclinical to clinical labeling contexts, where with human beings, you typically label people up to 1 to 2% deuterium oxide enrichment. That is a challenge when you when you're measuring the mass isotopomer distribution at the peptide level due to signal to noise issues, where you have very small changes, maximum theoretical changes in the M0 mass isotopomer. But if you're measuring the 13C isotopolog from the M1 mass isotopomer, M over Z space, you get very, very large changes in the 2H isotopolog abundance. So it's really actually pretty exciting stuff. So what I'm talking about here is looking at the ammonium ion isotope logs. So you take a peptide, you isolate it in the gas phase, you fragment it with high energy HCD fragmentation, and that gives you this resultant moiety. Basically, all of the deuterium oxide labeling is captured in this ammonium ion, and these can be then used as essentially reporter ions for quantitation purposes. And here's an example of what that labeling signal looks like for a proline ammonium ion isotope log, where this is from the M+1 peak in the from the proline amino acid side chain ammonium ion, where we have the 15N molecular species isotope log, the 13C, and the 2H. Interestingly, following metabolic labeling, you get a very large increase in the 2H isotope log. So this is labeling after 32 days, where this goes from its theoretical relative abundance of 0.02 to about 30%. And importantly, when you make this measurement, there's a fixed ratio of the N15 to 13C ratio. So this will never change as a function of deuterium oxide labeling, and it's ground truth, right? So when you take a sample, this should reflect how close you are to 8% for a proline ammonium ion isotope log. Tells you how well your instrumentation is doing. So it can be used as a built-in positive control for a stable isotope metabolic labeling experiment. And that can be, it seems like it's a very important quality control readout. Now, the methods that are being used, that I've been used, is a little complicated, but it's a PRM targeted assay on a Orbitrap, where we do 2 scans per target, where the first scan is at a low energy fragmentation, a standard energy fragmentation, to generate a peptide ID and do a peptide ID confirmation. And then the next scan would be with 100% NCE for the fragmentation energy, generating the ammonium ions. That can be used again as reporters for this isotope incorporation and new protein synthesis. So here's an example of a spectrum of a targeted peptide with the 25 NCE, and then the following spectra with 100% NCE, where if we zoom in closely for proline, valine, hydroxyproline, and leucine, we can very clearly differentiate the 2H isotope log abundance and the 13C from all of these. And what's cool after metabolic labeling is you have multiple reporter ions per peptide. So instead of one spectra telling you what's going on, or one scan telling you what's going on with the isotopic profile for the abundance, here you have a redundant set of reporter ions, which should be able to be used to increase your confidence for your analytic. Now, the recent application of this approach to measuring protein turnover synthesis in mouse tissues is as follows, is outlined as follows schematically, where we take mouse, we do an IP priming bolus, and then the mouse is maintained on deuterium oxide enriched water. Again, label gets incorporated during de novo synthesis of amino acids, and then we're going to quantify protein fractional synthesis by measuring the changes in the ammonium ion isotope log peak abundances. So mouse is labeled over a time series from 0 to 21 days. Here we're incorporating affinity enrichment of a bespoke target. subsequent digestion, and LC-MS in a PRM method. Now, if we were to use the Easy Spray, Easy Spray 1000 LC system, these separations or these runs would take approximately 50 minutes. Whereas on the Evosep, the method that was developed is a 5.6 minute method. So we get about a 10-fold decrease in time per sample analysis. And that's really, really important to me, because it makes a study like this more feasible, where a group had asked that we quantify protein fractional synthesis from 8 different tissues in mouse and 4 different groups of mice, male, female, wild type, in the genetic model of disease. So that, with controls built in, results in about 130 samples per tissue, which is approximately 10 hours of mass spec time if we're using the Evosep 1, versus about 100 hours on the Easy Spray system. And that's per tissue. So then if we do 10 hours per tissue times 8, so it's 80 hours versus 800 hours. 80 hours is definitely a feasible commitment to an effort. I wouldn't expect significant change in state in the instrument over time, over that short of time, whereas 800 hours, there might be a more significant drift and shift in platform stability issues. So I think that this coupling is really quite ideal for this kind of effort. Here's an example of what the isotopolog spectra look like as a function of label incorporation, where at time 0 you have very little 2H ammonium ion isotopolog abundance. It's basically the baseline theoretical amount, and as a function of new protein synthesis over 21 days, there's a graded and measured increase in 2H abundance, which is reflecting the new protein synthesis. Again, the 15N peak is relatively consistent, and then if you take the ratio of the 13C to N15 peak, this should work out to be somewhere near 8%, and the amount of deviation from 8% should tell you if you have issues, signal to noise issues within your data at that point in time. This is an example of what this fractional synthesis measurement looks like when we're looking across 4 different tissues just in the female group. The scatter, or just the spread of the data points at any one point in time, is fairly narrow. It's pretty good. And in fact, when we're looking at the comparison between the disease model and the wild type, tissues that are expected to show change do show statistically significant change. So it's a bit of a validation in that sense. Here's a heat map capturing all of the data, where we're comparing the fractional synthesis of new protein across the colon, lymph node, thymus, spleen, salivary gland, lung, heart, and muscle. And what we can see is the half-life of the protein ranges from 4 days to 24 days over these different tissues, which is really exciting if we are thinking about our bespoke target as the point of intervention, where we are trying to modify this protein's activity, what tissue matters. And we could model that explicitly, and we could also start interpreting what could be off-target effects if we know these differential half-lives, and we think we understand the mechanism of action for our drug. If it, say, were a compound that was driving degradation of protein or something like that, we would certainly have a better insight into on and off target effects with these data. What's also interesting is that we can combine the targeted and discovery data streams and look at the protein fractional synthesis of our bespoke target, which is the orange dot, across different tissues, and then comparing a distribution of protein half-lives between our disease models and between tissues to try and understand are the fractional synthesis rates of our target reflecting the tissue specific, or are they more target specific? And in this case, again, the half-life of the bespoke target looks to be quite discrete and distinct from the tissue itself, and is really reflecting the intrinsic biology and pathway dynamics within that tissue, and that was really exciting to see. Okay. And with that, I'd like to acknowledge all of the people that have helped with these efforts at GSK, and am open to answering any questions that you may have.
Yeah, thank you very much, Thomas. That was a very nice presentation, and impressive numbers of samples that you're talking about. So clearly something that's right down our alley.
Yep.
So if anyone has any questions, please type them into the chat box, and I'll be happy to forward them to Thomas. And in the meantime, Thomas, so how many samples have you actually run up until now? I mean, is this, I mean, this is really big efforts to produce this amount of samples, right?
Sure, it was probably around the neighborhood of 1,000 samples, and so typically, I mean, because of the nature of the sample preparation, the bottleneck was not the mass spec analysis, the LCMS. It was tissue homogenization and things like that, which was a good problem to have. It was nice to be able to rack up 130 samples, set them up, and have that data back, you know, after that 10-hour wait. It was really I think it really fits nicely into our data, our analytical stream.
Mm-hmm. And what about, I mean, so I guess you must, at least to some extent, rely on automation. Is this something that you can comment on, how this is set up from the crude sample to the MS analysis? What type of automation you have here?
So we've not incorporated more automation on the front end. However, you know, so we were processing samples in batches of 96, essentially, which is suitable for the Evosep tip, you know, stage tip capturing and sample introduction approach. However, you know, we are always interested in increasing the amount of automation that we're using in our efforts. So that's something that we're interested in, but had not used in this work.
Okay, thank you very much. So I do not see any questions in the chat box. So once again, I would like to thank you very much, Thomas, for this very nice presentation. Really much appreciated, and I enjoyed it a lot. I'd also really like to encourage everybody to visit our homepage, and especially our digital room, where this presentation or the recording of this presentation will also be made available. And then thank you everybody for your attention, and have a nice day.
Thank you.
Thank you, Thomas.